Hi everyone, many thanks for the organisers for the opportunity to present to you. Prescient is an ASX listed company with operations both in Australia and the US and commercialising and developing technologies from the US, funnily enough. But today I'm here to talk to you about Cell Prime, which is one of our programs that enhances adoptive cell therapy. A snapshot of our pipeline. I'm only able in the limited time today to speak about those ones in yellow, which is Cell Prime, but in the cell therapy space we also have quite a novel modular CAR system that is also cell agnostic that we've licensed from Penn, but I'll speak to Cell Prime specifically today. So Cell Prime can be used in two different ways. It is the same molecule, it's a small molecule, can be used in two ways during manufacturing. Cell Prime M for manufacturing, is a cell therapy enhancement for manufacturing to create a better phenotype of cell, a non-disruptive addition to the manufacturing process. And Cell Prime A being adjuvant, which can be administered to the patient, a completely different mechanism of action once it's given to the patient quite curiously, to again enhance existing and emerging cell therapies. So I'll speak about Cell Prime M first. Produces basically a better phenotype of cell. It is ready for the clinic right now. We have GMP material ready. We've got our own programs that we're progressing, but can be quite easily accommodated in third-party programs. We own the IP, and if anyone has cell therapy programs, CAR-T or otherwise, that thinks they could benefit from this, please come and chat to us. But basically, and this learned audience would know all about this, is that when a cell therapy product is administered to someone, it is basically a soup of cells. On one hand, you have naive memory T cells that have no activity but longevity, and all the way to the other end, effector T cells that have great potency but die out before the job is finished. And currently, there are way too many of these cells, and we know it's quite well documented now from third-party data that those patients that have quite stunning responses have a greater proportion of these cells, in particular central memory T cells. So it's all about trying to optimise the product that is given so that you can tip the scales and provide a product that has a greater proportion of central memory T cells. And at the moment, this is only done sort of batch by batch. Variability is still quite great, and you're relying on the green thumb, basically, of the manufacturer to try and optimise that. And basically, Cell Prime M pushes cells to this phenotype. And how does it work? It is basically dropped into the expansion stage of the manufacturing protocol as a very brief and temporary introduction to that. It is a 24-hour exposure, but we see effects within 15 minutes. Basically see gene expression very, very quickly. In terms of how that gene expression becomes, is translated to phosphorylation, we see within the first 15 minutes certain changes taking place already, and certain alterations in pathways after just 24 hours. And by then, certainly Cell Prime M has been washed out and is removed from the process. But you see alterations in pathways that influence the memory phenotype of the cell, and also increasing resistance, if you like, to oxidative stress, which is important to survive quite a hostile tumour microenvironment. The bottom line is this produces up to 50% more central memory T cells, which is highly significant, and it doubles the proportion of helper cells. And as those you may know, they not only have activity in their own right, but are strongly correlated with the longevity of CD4 cells. And not only do they have those sort of phenotypic benefits, but they also have greater chemokine expression, which is important for trafficking, which is especially important in solid tumours. It's one thing to have longevity, but getting to the sites in solid tumours is another big hurdle, and that's what this provides. So how do they work? So we used in this model we threw at it the hardest model that we could find, which was the Gottschalk model, which is the HER2 resistant model. And you can see by doing nothing to the actual construct itself, except for dropping Cell Prime M in briefly during the expansion step, you get a marked improvement in activity in a CAR T therapy that would normally be quite resistant. And here's what it looks like visually, untreated. You can see there's just basically no difference between the CAR T and the untreated population. But if you do nothing else except expose those exact same CAR Ts to cell prime M, then you get a marked improvement, all but disappearance of these highly resistant tumours. So how do these perform in vivo? How do these persist in vivo? You might think that once you administer these, they might revert to phenotype. This is cheek bleeds done a couple of weeks in, and you can see that the ratio is still very, very favourable. You get an increase, an ongoing and sustained increase in central memory T cells, and a decrease in that less desirable effector population, both in CD4s and CD8s in both cases. So this looks to persist in vivo some weeks in. So bottom line, this is a very easy way to integrate into existing cell therapy processes, be they CAR Ts or CAR NKs or any other adoptive cell therapy, we believe, to increase the phenotype. We have GMP material ready and delighted to speak to people about this. Very, very easy incorporation into programs. The same molecule can be used as an adjuvant, cell prime A. So again, given alongside a CAR T therapy, and it works in a very, very different way. I'll skip through this in the interests of time because I'll get to all of these points, but basically it increases in vivo expansion of CAR T cells 9-fold. Certainly for the CD8s and for the CD4s 6-fold. The differences, the improvements are highly synergistic with cells that have actually been made with cell prime M. So again, a marked increase in vivo expansion, and that results in significantly more cells penetrating the tumour. So again, 3-fold and 4-fold more respectively for CD4s and CD8s. So I guess 9-fold more soldiers deployed to the battlefield and up to 4 times more soldiers actually breaching those castle walls. So that's significant. And also, regardless of the modality, this is obviously highly important, the ability to reduce Tregs by 2/3. And as you know, they are important in sort of blunting the immune response. So to be able to do all of those things, you can see why we're getting such a synergistic effect using cell prime M and cell prime A together. And this is again demonstrated here in that exact same model, the HER2 model that I described earlier that is highly resistant. You get a benefit in cell prime M, you get a benefit in cell prime A, and when you use them together, you get a highly synergistic benefit in this highly resistant model. So again, cell prime A is easily incorporated into clinical trials as well. There are groups speaking to us about adding this as an additional arm onto their existing studies to either have patients who would otherwise be resistant to CAR T therapy respond a little bit better, and for those therapies, especially in solid tumour programs that might be more marginal programs, to really move the needle on that without any changes to the construct itself. It's both this molecule in both its forms. It's very well described. There's clinical experience with it. There is a robust regulatory package, and the idea is for us to have an umbrella IND that others can reference relatively easily. So that's it basically. It's a very quick snapshot of what we're doing with plenty of time spare, a manufacturing enhancement to really increase the phenotype of these cells to last longer and kill for longer, and also as the adjuvant therapy to really prime the tumour microenvironment, increase in vivo expansion, and have these cells actually penetrate the tumour and have the tumour microenvironment more conducive to these cells doing what they need to do once they're there. So that is that, and I got through that much quicker than I thought I would. So many thanks.